Now move on to the action potential proper. Action potential. Action potential, you know, I previous part I mentioned earlier, I mentioned that action potential is the feature of the excitable cells, that is nerve and muscle fiber, nerve and muscle fiber. So action potential is defined by many, action potential is defined by many authors, somewhat different, the definition are somewhat different, but the theme is nearly same of all the definitions of all the, you can follow anyone any definition, and I included the definitions in notes also. So there are 4 definitions of 4 different authors, and I would like to write the definition of Guyton and Hall. Guyton and Hall, 13th edition, 13th edition, 13th edition. According to this book, or according to them, action potential is action potential action potential is rapid, action potential is change in rapid change in membrane rapid change in membrane potential that spreads it spreads along nerve or muscle fiber, along nerve or muscle fiber, allowing the electrical signal electrical allowing the electrical signals to be transmitted. This definition is by Guyton and Hall, 13th edition, physiology. So what is the main point? It is a rapid change in the membrane potential that spreads along the nerve or muscle fiber, that is excitable cells, all the way, spreads all the way or throughout the fiber, allows the electrical signals to be transmitted. The function is transmission of the electrical signals, and you know, in nervous system and muscular system, the signals or message is transported as electrical signals, and the desired action is achieved. So this is very comprehensive definition, and there are many other definitions also. So if you write giving the reference, I said in previous lecture also, if you write giving the reference, the examiner will get impressed by your answer, that you are writing some authentic thing. And again, you can write in your own words also. So let's simplify it. I simplified from other definitions, and by the definitions also I simplified form. The action potential is action potential is rapid and sequential rapid and sequential. Rapid and sequential change in membrane potential. Rapid and sequential change in membrane potential that travels along nerve or muscle fiber, nerve or muscle fiber. uh, Here I would like to add some point in membrane potential. Membrane potential here you can add which is produced by application of threshold stimulus. Remember, this action potential follows all and non rule, and by that rule it will generate and propagate only if the stimulus is threshold. If the stimulus is threshold stimulus, threshold stimulus that travels along nerve or muscle fiber and transmits electrical signals, electrical signals. This is more comprehensive and more collective, while rapid and sequential, not only rapid but is also is sequential, that the transmission occurs in sequence, or change in the membrane potential occurs in sequence. Like first is in polarized case, polarized or resting phase, then it will depolarize, then again it will re repolarize. That is resting phase, depolarization and repolarization, and this phenomena repeat so forth. So this is in sequence. So hence called sequential change in the membrane potential. Sequential change, and that travels same thing along throughout the membrane of the nerve or muscle fiber, and function is transmission of the electrical signals. So you can write in your own words, I write in my own words, that action potential is rapid and sequential change in membrane potential which is produced by application of threshold stimulus that travels along nerve or muscle fiber and transmits electrical signals. And there are many other definitions also. You can follow any of the definition. Now talk in detail action potential. After definition, you will be asked to write phases or ionic basis of the action potential. So let's jump into the phases of the action potential. Phases of the action potential. Phases of action potential. Phases. Let's draw a figure. for better visualization. This is the potential, and I will talking in reference to the of neurons, neuron potential, not muscle like a shoulder or cardiac muscle. I will at a time I will talk in about the of neurons action potential, now large nerve fibers, in large nerve fibers, and this will be your millivolt potential, and this axis will be of time, time in millisecond, time in millisecond, millisecond. So um, somewhere here, let us suppose somewhere here is minus 70 millivolt, somewhere here is the 0 millivolt, and here is the plus 30 or 35 35 millivolt, and time here will be, let us suppose time here is 0, 0 0.1, 0 0.2, 0 0.3, 0 0.4, 0 0.5, and etc. Time is in fraction of the second, even in fraction of the millisecond, because this signal should be transmitted very rapid, very rapid. Uh, let's here threshold stimulus if applied, the potential will rise, it will reach to the positive potential and then will come back, and next potential will be generated. And next action action potential will be same, in same manner will be generated and transmitted with same amplitude. Once it is established, once it is generated, the amplitude will not change throughout its pathway, throughout its pathway. This this length, this dimension is you call as the amplitude. What I want to show is here that let us suppose somewhere here is threshold potential, threshold potential, threshold potential, or you call it as the firing potential, firing potential here 0 and here plus 35 equivalent millivolt. This is your spike potential, spike potential, or you call it as spike potential or overshoot, overshoot, overshoot. Here this is okay, this is depolarization after threshold potential is reached, fully depolarization occur, so you call it as d. All this, when potential tries to rise towards positive, that phenomena will be the depolarization, depolarization, and this is your once depolarized it should be repolarized. So repolarization, repolarization, and this will be hyperpolarization, hyperpolarization. Why this, how this occur, I will show that also, I will talk about that also. And this opposite of this overshoot, you call it as the undershoot, undershoot, undershoot. So see here, one cycle of the action potential is established and terminated within fraction of milliseconds, that is fraction of within 0.2 to 0.3 millisecond. And if you also want to quantify the time, this depolarization occur 1 by 10 thousandths 1 by 10 thousandths of a second. Remember, this will be your 0.000 nearly right near 1 second, and repolarization in also in that same time, in that fraction of the second. So how rapid this process is, you can see. And you should, in exam you should draw the figure also of this action potential. You should illustrate with the figure action potential. Action potential. So first phase here will be, this is the resting membrane potential of the large fiber minus 70 millivolt. Then first will be resting membrane potential, or simply called resting stage. Resting stage before nerve fiber is stimulated or excited, before excitation or stimulation. Excited, at this condition the action potential is not generated and not transmitting. The potential will be minus 70 millivolt, that is minus 70 millivolt more negative inside than outside. The potential will be, this is resting membrane potential. Now the next phase will be your depolarization stage. Depolarization, now here depolarization and action potential will occur only if the threshold stimulus is applied. Depolarization will may occur at some point and return to back without generating the action potential if the stimulus is sub threshold. So depolarization how will occur? Due to diffusion of sodium ions inside the membrane through initially inside the membrane. The initially or till threshold potential or firing potential diffusion occurs through diffusion of this sodium, diffusion occurs through mechanical mechanically gated or ligand gated sodium channels, ligand gated sodium channels. Remember, from beginning the diffusion of sodium will not occur through the voltage gated sodium channel. At first, till the firing potential is reached or threshold potential is reached, till then the diffusion of sodium will occur through the mechanically gated or ligand gated sodium channels. Mechanically gated means what? If you give any mechanical stimulus like pressure, fine touch, this is your mechanical stimulus. In that case, the initial diffusion will occur through the mechanical gated channels, and ligand gated channels like at synapse at the synapse at the let us suppose this is the axon of one nerve and this is dendrite and this is cell body of the under nerve, then in that case these axon terminals, once axon potential is reached here, the communication is done by when these axon terminals release the neurotransmitter by mechanism, neurotransmitter, neuromuscular or synaptic transmission. I will talk in other part that here neurotransmitter will occur and this neurotransmitter will stimulate this postjunctional membrane, and here somewhere here called axon hillock, and that axon this influx of the or binding of the neurotransmitter and postjunctional membrane will again open the ligand gated, here will open the ligand gated sodium channel, and this ligand gated sodium channel, through this ligand gated sodium channel, once the threshold potential is reached, the action potential will generate here axon hillock, and then again will be transmitted to other fibers or the effector organ tissues. So here initial entry is through the ligand gated sodium channel at synaptic transmission. synaptic transmission. So, and again I would like to mention that at normal physiological condition there is again very important viva and MCQ question. MCQ question is that at normal physiological, I will deviate from this for a brief period of time. At normal physiological condition, normal physiological condition, like in that case synaptic transmission condition, action potential will generate at axon hillock. This is just immediate to the in the vicinity of the this cell body. Here is you call as the axon hillock, and this will be your axon. So axon hillock, the action potential will generate here and will be transmitted. Axon hillock, and the reason why occurs at axon hillock, because due to more more or greater concentration of more or greater concentration of voltage gated sodium channels than other parts of the axons. More voltage gated sodium channels will be here at axon hillock. So the action potential will generate here. Where occur and why, the reason is here. So this is again your important Viva and MC question. Now the question again arise. Let's suppose I this is, let's suppose, cell body and this is axon. If I give the threshold stimulus here at this point, then or away from cell body or away from the axon hillock, then at that time also the action potential is generated at the axon hillock. What do you think? Will the action potential again generate at axon hillock? No. Answer is no. If the stimulus is away from the axon hillock, the action potential will generate at the site of stimulus and travels or transmitted bidirectionally. And again this is the important feature of the action potential, that action potential once generated, it transmits bidirectionally, that is in either direction from the site of the stimulus, this point. And again one more question your examiner will ask, that if is transmitting towards the cell body and dendrites, then the transmission further occurs from the to the other axon terminals from that also occur? No, normally it does not occur, because the mechanism, neurotransmitter vesicles, calcium channels that facilitate or present at the axon terminals is not available here. So the action potential will die out here, but in this direction will transmit if there is other neurons or muscles, or will stop if the effector organs is there. So at normal condition, the transmission is mainly exodendritic, that is action potential transmits from axon terminals of the one neuron to the another cell body or dendrites of the another axon on the nerve fiber. This is major pathway. And very rarely there is also the dendroaxonic synapse that is synapse between the dendrites and axon, and in very this is found in very rare cases, and in that case the transmission will occur from this dendrites to the axons also, but this is very rare. Very rarely this transmission is found in the body. So question, what will be the question? That bidirectional transmission from the side of the stimulus, it will transmit normally towards the axon, axon terminals, and then to other neurons, and this by in this direction, once it come to the cell body, it will die out except in dendroaxonic transmission synapses, which is very rare. So again I would like to be back in the depolarization. Depolarization. Depolarization due to more concentration of the voltage gated sodium channels. Sodium channels that is present at the axon hillock. Axon hillock. Now, uh, yes. Then what is the firing potential or threshold potential? Threshold potential for this nerve fiber, the threshold potential will be minus 55 millivolt. Threshold or firing potential will be minus 55 millivolt. That is how action potential is generated. Again, let's talk. Let's suppose this is nerve fiber. Here is positive charge, negative charge, and same will on that surface also. The surface also. Once the stimulus, threshold stimulus is applied, what will happen? These sodium ions will diffuse inside the membrane, and there is the higher concentration of the potassium inside the membrane. You know also that once the stimulus causes the influx of the sodium, the efflux of the potassium will try to neutralize this change in the membrane potential. Will try to neutralize the change in the membrane potential, but there is some limited capacity of everyone that if the sodium diffusion is the diffusion of the sodium is greater enough than the diffusion of the potassium, the action potential will generate. And if the stimulus is threshold, the influx of the sodium ions will be neutralized by the efflux of the potassium, and the action potential will not generate at all. So this is the practical thing how the action potential is generated, and the theoretical value is minus 55 millivolt. So conclusion you can draw is that if the diffusion of the sodium is greater than the diffusion of the potassium, then action potential will generate, or if the membrane potential change means the membrane potential change by greater or equal to plus 15 millivolt, greater or equal to plus 15 millivolt, the action potential will generate. So here plus 15 milli equivalent is in this potential, this 15 milli equivalent positive potential uh, increases the potential in positive direction from minus 70 millivolt to the minus 55 millivolt. So this is the mechanism how action potential is generated, and this mechanism I also explained in homeostasis, positive feedback mechanism, and this external potential is generated by the positive feedback mechanism. Remember, you can refer the homeostasis feedback mechanism. And again, so here it at first the diffusion of the sodium will occur through the ligand gated or mechanically gated channels. Then if the when the threshold is reached, when the threshold potential is reached, this will cause the conformational change in the voltage gated uh, sodium channel. I will show in the ionic basis. Conformational change, this will lead to the conformational change in the voltage gated sodium channel. Conformational change in voltage gated sodium channel activate or open the voltage gated sodium channel, and rapid and in a huge amount rapid diffusion of the sodium will occur through this voltage gated sodium channel inside the membrane, and the potential will rise till the positive value and in large number of fibers it reaches the positive value, like plus 30, 35. But in some small nerve fibers, or in nerve fibers of the central nervous system, this potential merely rises to the positive value, or merely rises to the zero, and action potential depolarization will stop and repolarization occur, and the nerve impulse is transmitted. So this is the depolarization stage. Now let's move on to the repolarization. Repolarization. Once the membrane will get depolarized, it should again be repolarized. Stage 3 will be repolarization. Stage. Repolarization stage. How the repolarization will occur? Once the threshold potential is reached, that during this period the influx of sodium will occur through the mechanically gated or ligand gated sodium channel, and then after the threshold potential, uh, voltage gated sodium channel will open, and influx of the sodium rapid influx of the sodium will occur, that lead to the overshoot of the membrane potential. And after the fraction of few millisecond, after the fraction of few millisecond repolarization, uh, repolarization, as soon as depolarization cause the overshoot of membrane potential, potential, the sodium, the voltage gated sodium channel will close, voltage gated sodium channel will close, and the potassium diffusion of the potassium through the membrane will occur to neutralize the depolarization potential, and along with that the voltage gated potassium channel will also open very soon, and the membrane potential rapidly brings like diffusion of potassium and uh, diffusion of potassium mainly by voltage gated potassium channel will cause the or lead to the repolarization. Repolarization along with diffusion of potassium ions through leak channel, through potassium leak channel. The major efflux occurs through this voltage gated potassium channel, and simultaneously the potassium will also leak to outside the membrane through the leak channel, since the permeability of the potassium is very high in comparison to the permeability of the sodium channel. Hence through leak channel, and within fraction of millisecond membrane potential, within fraction of millisecond membrane potential returns toward resting membrane potential. Now talk about the hyperpolarization. Phase 4 will be, let's write here, hyperpolarization. Hyperpolarization occurs during this repolarization stage. The potassium channel will remain open few more fraction of milliseconds after it returns the membrane potential towards resting membrane potential, and that that opening below the resting membrane potential, that opening lead to the hyperpolarization. That during this phase, once RMP is reached, at same time the potassium channel will not close and remain open for more fraction of few milliseconds, and that leads to the like right that uh, delayed opening of potassium channel will lead to the hyperpolarization. That is, potential will decrease even more than minus 70 millivolt. So this will occur by the delayed opening of the potassium channel. You can see here, this is the depolarization, and this depolarization is also called as the undershoot. And then after a few milliseconds, the potassium channels will close and diffusion of potassium will stop, and hyperpolarization will be prevented. And after that the sodium potassium pump will get activated, and the disrupted concentration of the potassium and sodium inside and outside will be re-established by the sodium potassium pump, sodium potassium active ATPase. And this uses, as also mentioned earlier, this uses this re-establishment alone by sodium potassium pump in the nerve fiber uses 60 to 70% of energy, of total energy used by this nerve fiber. So remember how important the sodium potassium pump is here. Now this ends here the stages or phases of the action potential. We now will move on to the ionic basis. Sometimes phases and or ionic basis is also asked in your SAQs. So the phases or ionic basis are not very different thing, but just some point is different. The mechanism or the action is same for both ionic basis and phases. Bold. Now talk about the ionic basis. Ionic basis of action potential. Ionic basis of action potential. The here will be first phase will be influx of sodium ions. Influx of the sodium ions. At depolarization, influx occur of the sodium, and at repolarization efflux of the repolarization efflux of the potassium will occur. So influx of sodium channel initially till threshold threshold or firing potential potential initially till threshold potential is reached the influx will occur. Influx of sodium will occur through mechanical or ligand gated sodium channel. Then once this is reached, after threshold potential is reached, influx will occur through influx of sodium will occur through voltage gated sodium channel. And all this mechanism will cause the lead to the depolarization. So first phase is the influx of the sodium ions. Now next will be efflux of the efflux of the potassium. By voltage gated potassium channel mainly, and diffusion of potassium through potassium leak channel. This will lead to the repolarization and may cause hyperpolarization due to delayed opening of few millisecond, fraction of few milliseconds, delayed opening of these channels you can see here hyperpolarization is causing. Then next will be the re-establishment by sodium potassium pump. This sodium potassium pump, this pump sodium potassium 80 phases, sodium potassium 80 pH system. This pump re-establishes the resting membrane potential as well as uh, correct ionic concentrations inside ionic concentration of sodium and potassium outside and inside of the membrane, outside and inside of the membrane, because once since here during the depolarization phase sodium ion is getting influxed inside the membrane, and to counter this depolarization during the repolarization period, potassium ion will get effluxed. Then by this mechanism electrical neutrality is maintained that here it will lead to the positive potential, will cause the positive potential inside, and to neutralize this positive potential, potassium will get effluxed and the normal negative potential will be established inside, but the sodium ion concentration will increase inside the membrane and potassium ion concentration will increase outside the membrane during this action potential cycle. Then this will be again the concentration will be maintained at initial phase, that is normally the sodium is higher concentration outside and potassium is higher concentration inside, and here this mechanism just reverses the concentration, and this reverse of the concentration will again will bring back to the normal concentration, original concentration of sodium and potassium inside and outside by this sodium potassium pump, sodium potassium ATPase mechanism. This will lead to the efflux of the 3 sodium and efflux of the 2 potassium at each time by using the ATP by using the ATP, and it will break into ADP plus inorganic phosphate. The enzyme will be sodium potassium ATPase, sodium potassium ATPase, sodium potassium ATPase. So this is the ionic basis of the action potential. Now in next part I will talk about the its clinical correlation, that in which fiber the action potential, uh, which type fiber action potential uh, travels faster and in which travels slower, and how drug can be applied to reduce this nerve impulse transmission like LA, and the anesthetize the uh, nerve for the nerve or muscle for the minor surgeries. So all I will relate in next part. Hope you enjoyed the lecture. Thank you for watching.